There are three types of neurons within our biological brain that have key roles and responsibilities in generating our brainwaves. These cells are called pyramidal cells, fast spiking cells or FS cells, and non-fast spiking cells or NFS cells. While the NFS and the FS cells are inhibitory, the pyramidal cell are excitatory. Because both NFS and FS cells are inhibitory, they actually compete with each other. So NFS is non-fast spiking, FS is fast spiking cells. They are inhibitory, meaning to say that they inhibit the brain to become excited. So sometimes when they work with each other, they actually compete with each other to inhibit the brain from becoming excited. For example, when we are resting, the NFS or non-fast spiking cells work together with pyramidal cells to inhibit our excitatory systems, and as a result they create more beta waves, and this NFS also inhibits the FS cells. On the other hand, when we're active, the fast spiking cells become activated. They work together with the pyramidal cells to create gamma waves, and they inhibit the NFS cells. In this particular study, these researchers set out to investigate what are the underlying mechanics when it comes to different brain waves and these different cells and circuits in our brain. If you remember, we talked about working memory in the past and how crucial it is to our survival, because although its capacity is very limited, it's definitely crucial to our day-to-day interaction, because we need to remember things in a short period of time without the stimuli being present, and that is what our working memory is. One of the findings showed that there appears to be a division of labor between fast spiking and non-fast spiking when it comes to the dynamics underlying working memory between the cells and the brainwaves. Furthermore, the interaction between the pyramidal cells and the non-fast spiking cells appear to generate beta waves, whereas the interaction between the pyramidal cells and the fast spiking cells appear to generate gamma brainwaves. What they noticed was that before showing the cues to these participants, the pyramidal cells activation was low, but the non-fast spiking activation of these cells was high. Hence, the beta waves activation was high. Whereas when they showed the presentation or the cue to the participants, the pyramidal cells activation got high instead. That led to the fast spiking neurons becoming activated, which also then led to gamma brain waves being activated and elicited more. The same phenomena may be also the underlying mechanism when it comes to attention or perceptual decision making in humans, where the gamma waves are increased and the pyramidal cells basically recruit more and more fast spiking neurons. If you remember, also in the past we talked about how gamma waves are very important when it comes to sensory information, and it appears that the activation of gamma in the sensory cortex has a lot to do with it somehow encoding sensory information into these cells, into our brain. To illustrate, sensory information increases the excitement in our sensory cortex, which then increases the activation of the fast spiking neurons, which then contributes to increased elicitation of gamma waves, increased activation of gamma band frequencies in our brain. As we talked yesterday, in essence, brain wants to always keep its stability, its equilibrium. So although there is a lot of excitation and inhibition in our brain be it at the neuron level or brainwaves level, there's always an underlying mechanism where the brain wants to deal with this excitation and inhibition, but then go back to normal, basically, be a little bit more stable. And for that reason, we also have these synapses between the neurons or between the brain cells that are short-term. And so plasticity of these connection of these synapses when they are short-term also plays a huge role to help the brain have these excitation and inhibition, but they're all short-term. They are not long-term. That way, the brain can go back to its stable and equilibrium state. And so the plasticity of these short-term synapses allows the brain to be fluid and react differently to whether a neuron is excitatory or inhibitory, whether a brainwave is excitatory or inhibitory. And so the brain constantly seeks for a balance between excitation and inhibition, because imbalance between excitation and inhibition in the brain can either cause us to have false memories, or the inhibition can cause us to be forgetful. For example, when these short-term synapses are excited for too long between the pyramidal cells and non-fast spiking cells, then we have too much excitation and false memories may be created. On the other hand, too much inhibition can cause us to forget because we will have harder time maintaining information. And so for the brain to create and remain its balance, there has to be a short-term plasticity among these different cells, especially when it comes to the pyramidal cells with fast spiking versus fast spiking cells, because that is the plasticity of the short-term synapses that helps the brain to be flexible enough to go back and forth between exhibition and inhibition to create this balance. And all of this gives the brain the capability to be able to function properly when it comes to its working memory, for example. But the very interesting part of this study was that, again, scientists are combining now the physical with the non-physical brain in order to understand, in this case, for example, the underlying mechanisms of working memory, because as we've talked in the past, so far, the majority of the time in the past century, the neuroscience community has been primarily focused on the physical brain, meaning to say the neurons spiking, etc. Not that there is anything wrong with that. We have to have them, but we always tend to miss this sort of holistic way of looking at the brain because we kept forgetting and at least not including the brain waves in our studies. But these type of studies show that we are awakening more and more to this type of approach in the neuroscience community.